Hello everyone, in today's video I am going to explain about the structure of a dicotyledonous seeds. In previous lecture I had already explained about the structures, or we can say the types of seed on the basis of the number of cotyledons. That was the monocotyledonous, and the second types, that is the dicotyledonous. So in today's video the topic is about the general structure of dicotyledonous seeds. So here is the diagram of a dicotyledonous seed. This is the external structure, in which the outer covering, that is named by the term testa. Inside that there is a micropyle, and the centric region, that is the hilum, which consists of ovary, and inside that ovary the ovule are attached. So there are two cotyledons. Now this is the diagram of an open bean seed. These open bean seed consist of two cotyledons, which is the outer covering present, that is termed by the name seed coat. Inside that there is a embryo, which consists of a radicle and a plumule, and the upper region, that is hypocotyle, and the second region, that is epicotyle. So this was about the diagram of a external structure of a bean seed and the open structure of a bean seed. So we will look at the structures of the dicot seed in brief. So starting with the structure of these dicot seeds, the first one is that dicot seed are basically a kidney shaped structure with a convex and a concave side. Basically these seed are basically structures are sometimes the convex and sometimes the concave side. So now starting with the structural components of this dicot seed, the first one, that is the seed coat. This seed coat consists of the outermost hard covering, that is termed by the name testa, and inside this testa there is an inner thin layer, or we can say inside this testa there is another secondary layer that is present inside, that is called by the name tegmen. So what are the purpose of these both layers? These both layers protect the inner delicate part of the seed from any physical injury or from any attack or pathogenic attacks. Now the second structure, that is the hilum. As I have already shown in the diagram, that is the centric region of a bean seed. This is hilum. Now this hilum is basically oval, light shaded, or close to hilum, or that scars represent the spots, white spots. So where ovules was attached to the ovary through placenta. So in this hilum region the ovules are basically attached to the ovary, as we have already seen in the structure of fertilization process, in which there was another structure, that is placenta, a tissue that helps to join the loculus. So the third one, that is the micropyle. So these micropyle are basically tiny pores situated close to the hilum, and these are basically, if we talk about the function of these micropiles, these are helps in the absorption of water for the seed germination, and also sometimes allows the diffusion of gases or respiratory gases for the seed, sometimes the exchange of gases of carbon dioxide inside and outside the seed. So the fourth one, that is the cotyledonous, or we can say the cotyledons. These cotyledons are two thick seed leaves that store the reserve food, or we can say food materials, for the nourishment of the growth of the embryo. As I have already seen in the diagram, there is an embryo which will grow into a shoot region and in root region. So the last part, that is the embryo. So this embryo is attached to one of the cotyledons, either the left side or either the right side of a cotyledon. So these embryo consist of the two types. The first one is the radicle and the second one is the plumule. The radicle, which later forms the root, and the plumule, which later form the shoot. You will able to observe it in a sprout. The structure of the plumule. The plumule are basically the outer region that forms the shoot in the plant, or we can say the germinating part of the seed, basically the plumule, and the root region that is named inside the seed, that is termed by the name radicle. So this was about the structural components of a dicot seed. In the next part of the video I am going to explain about the structure of monocotyledonous seed. Till then, thanks for watching.